All right, everyone. I don't want to get bad reviews for going over time, so we're going to get started on time. My name's Jay Freeman. I'm a professor of pediatrics at Nationwide Children's Hospital, clinical pancreatologist. Got some support in the back of the room. Thank you. Appreciate it. I'm joined by Allison Cohen as my co-moderator, who is an associate professor of internal medicine at Pittsburgh and a lipid metabolism biologist, making her much smarter than me. So any complicated questions go to her.
Thanks a lot. All right. Are they going to pull up? Can they pull up?
You have to go take it.
Okay, sorry. I just want to set the casual vibe for this talk. Can you?
Let's see what we got.
Can they pull up our presenter slides? Understand what's going on here. Aha, thank you. Thanks. All right, so welcome to our session. I just have a couple disclaimers, the disclosure slides, and then want to underscore our objective for this session, which is to talk about both the clinical and basic science part of gastrointestinal manifestations. We're going to start in the pancreas, move on to the small intestine, learn a little bit about the microbiota, and then focus on the gut-neuro axis. And I'd really encourage everybody to ask questions at the end of each talk. There's also an opportunity to submit questions through the app, and then I'll read those aloud. So don't be shy. Come up to the microphone and ask a question. Okay, so I would like to introduce our first speaker, Dr. Yunksha O'Malley, an associate research scientist at Iowa. And I'll let her get started. Oh, thanks.
Thank you all. Be here. The title is, do you hear me? Yeah, okay. The title is CFTR inhibits cell division to suppress pancreatic duct cancer cell proliferation. There's no conflicts of interest related to this presentation. What is the relevance to study the connections between CF and pancreatic cancers? This publication indicate that people with CF, after follow 3 to 7 years, GI cancer risk increase 6 to 19 folds. Pancreatic cancer increase 6.8 folds. Here's publication. This blog is pancreatic cancer. Here's a statistic. Pancreatic ductal adenocarcinoma, in abbreviation PDAC. In 2023, PDAC was the fourth most deadly cancer in America, accounting for 8% of all cancer deaths. In 2024, English study indicate that PDAC had lowest 5-year survival, 8.3% out of all malignancy. Metastatic PDAC only 5 to 11 months survival. A 2024 study found that germline CFTR mutations may exist up to 12% of all PDAC tumors. Pancreatic tumors are often resistant to cancer therapies. The challenges to study pancreatic disease in CF: inability to obtain serial human samples. It's invasive, it's impractical. It's difficulty to grow and propagate in vitro models. Rodent models have mild pancreatic disease. In 2008, this University of Iowa CF research group published this paper and they create the pig CF models. Pig lung anatomy, airway structures, and immune system are similar to those of humans. But why use pig model for research of pancreatic CF disease? Dr. Alia Uch in Iowa have studied pig models since 2010. They discovered CF pancreas tissues have inflammation, acidic concentrated secretions, ROS generation, oxidative stress, fibrosis, tissue remodeling, and increased cell proliferation. These characteristics also resemble human CF pancreatic disease. The hypothesis is higher pancreatic ductal adenocarcinoma, PDAC, risk in CF may be due to uncontrolled cell proliferation, which is modulated by lack of CFTR. We generate pancreatic ductal epithelial organoids. The pancreas is minced, washed, enzyme digested, and washed, and then embedded in matrigel and cultures. 2 to 6 passages is used to measure PDAC characters, PDEO characters, and functions. The median is lab-made, and based on the reference of these articles, pig pancreatic duct epithelial organoids demonstrate polarity with F-actin in the apical side and sodium-potassium pumps on the basolateral sides. They express epithelial and duct markers and well-known transporters, including CFTR, NKCC, MBC, and NHE. No acinar and islet cell markers. CF organoids compared to wild type cells the organoids are small and clusterings. The functional assay shows wild type organoid swelling is stimulated by chloride and phosphorin in a dosage-dependent manner. CF organoids have no response. Wild type also have CFTR dependent bicarbonate secretions. To study cell division, we culture the cell from organoids to monolayers. The monolayer cells demonstrate the markers of epithelial cells, beta-catenin and ZO1, and also SUX9. This is our experimental design and method. Pig organoids culture in the monolayers, and we also include 2 cancer cell lines, CFPAC1 with delta fibroid mutation, and PANC1, which is a non-CF wild type cancer cell lines. After certain days of culture, 3 major assays were performed. Count cell to generate cell growth curve, PI stain and flow cytometry to analyze cell cycling phases, and gene sequencing to analyze gene expressions. Pig CF PDAC have much higher, these y-axis cell numbers and x-axis as culture days. CF cells grow much faster compared to wild type, with doubling times of 1.8 days compared to the wild types in 2.2 days. Then we ask if we can increase CFTR expression, would that inhibit cell proliferation? We transfect adenovirus contains CFTR 25 and 50 MOI and also GFP accordingly. CFTR overexpression inhibits cell proliferation and also increase CFTR mRNA expression. Please notice the 25 MOI mRNA expression is reached the same level as wild-type control samples. With lower MOI, 1, 5, 10, 25 also inhibits cell growth. Cell doubling time curve also shows dosage response with CFTR expression and compared to GFP. This is human tumor samples, and here's the cell samples. In human tumors, CFTR mRNA expression is lower compared to the normal tissues. And cancer cell line also express much lower mRNA, CFTR mRNA, and all non-detectable mRNA expression compared to CFTR wild-type samples. Then we increase CFTR expression in cancer cell lines. This up panel is CFPAC1 cells and bottom's PANC1 cells. Increase CFTR expression with 25 MOI and 50 MOI and inhibit cell growth compared to GFP. And also increase mRNA expression, 25 and 50. The similar result also happens in PANC1 cells, 15 MOI and 25 MOI. And mRNA expression also increased after transfections. Then we ask if CFTR modulators also inhibit cell proliferation. The answer is yes. The modulators, here's the letter shows the combination of modulators. With combination of ETI, cell proliferation is inhibited, but not if you apply separately, applying. This phenomenon also happens in cancer cell lines. With CFPAC, ETI inhibits cell proliferation, PANC1 also inhibit after ETI treatment. Then we perform cell cycling phase analysis with purpurium iodine stain the cell nucleus and go perform flow cytometry assays. Cell cycle progress goes through 3 major phases, G1, S, G2, and mitotic phase. 3 types of cells were used: PIC, CFPAC1, PANC1, and G1 phase, S phase, G2 phase. Y-axis is percentage of cells. X-axis is days, 1 day, 3 days, and DMSO as control, ETI as treatment. In 3 cell type, G1 arrest happens, PIC cells, CFPAC1 cells, and PANC cells on day 3. PANC cells also can be detected in day 1. What about the RNA expression? 3 cells, PANC, CFPAC, PANC1, and PIC cells. G1 phase, S phase, S2 phase. DMSO, ETI treatment. Overall, G1 phase and S phase in cancer cells all down-regulated. But in the pig cells, in the PDAC cells, genes are upregulated. Analyze positive regulators genes in this category. Cancer cells all downregulate, and pig cells upregulate. Negative regulators have no change. Then question will be, what about cell signaling pathways? Some reports indicate Wnt beta-catenin pathways are upregulated in the CF cells. These are Wnt beta-catenin signaling pathways. Let's focus on the membrane component. Wnt, FZD, and LPR. And here's the raw data of Wnt, FZD, and LRP. In summary, membrane components, Wnt, FZD, and LRP6, all down-regulated, and PORCN down-regulate in pig cells, inhibit up-regulate in the cancer cells. In summary and conclusion, loss of CFTR is linked to increased proliferation of primary pancreatic duct cells, which can be suppressed by enhancing CFTR expression or activities. CFTR expression is downregulated in PDAC, and restoring CFTR reduces cell proliferation. CFTR modulators inhibit cell proliferation by inducing G1 phase arrest in the cell cycling of primary pancreatic duct cells and PDAC cells. CFTR modulators downregulate key regulators in Wnt beta-catenin signaling pathway. Further studies are needed to study the impact of CFTR modulators on pancreatic tumorigenesis. There are many people to thank for this research, and Dr. Alia Uch's lab, CF research group people, is over the years students and faculties involving, and millions of thanks to University of Iowa CF lab, and thanks for the funding of Children's Miracle Network. Thank you very much.
Thank you for starting us out with a great talk. We have 2 microphones here in the middle of the room. As mentioned earlier, don't be shy. I'd love to have a great conversation.
I'm going to start with a question. Do you think that the effect is due to a change in pH?
There are some indicators in my other experiment. But I haven't really measured. I just take some photos. Yes.
Okay.
Good question.
Thanks.
Following questions. Do you think it's happening because you're changing the calcium level into the cell? Because usually this CFTR modulators change the calcium. Once you affect the calcium, you will reduce proliferation and not this thing.
I'm sorry, we haven't measured the calcium. It will be interesting to measure it next when we work on other area. Yeah, there's many things to work on after the modulators of treatment.
All right. Well, if there's no other questions, thank you very much. We'll move on.
Thank you.
All right, next we will be hearing from Derek Host. Derek is a student in Mitch Drum's lab at Case Western University, and he's going to be talking to us about metabolic shifts in the CF mouse small intestine and potential transcriptome drivers of glycolysis.
So thank you. My name is Derek Host. I'm a PhD candidate in Dr. Drum's lab at Case Western Reserve University. My research is talking about the transcriptional mechanisms behind the glycolytic upregulation we see in the CF small intestine. So I have no disclosures. So just some quick background on the kind of the CF intestinal manifestations we see. We have some obstruction, meconium ileus, Dios distal intestinal obstruction syndrome, dysmotility, some small intestine bacterial overgrowth, increased risk of CRC or colorectal cancer, and constipation. We may have many underlying mechanisms, such as the chronic inflammation, dysbiosis, or impaired barrier function. And more importantly, the G542X mouse model that we use recapitulate a lot of these intestinal phenotypes that we see. So starting on with, we had an observation by Aura Perez who showed that in both fed and fasted states, our G542X mice had lower levels of plasma glucose, but we didn't quite know where that glucose was going. So Alex Lesser from the lab conducted radioisotope labeling of wild type and CF mice, and we can see here the larger signal in the intestinal region, showing increased uptake from the circulation into the intestines, and no significant difference in the heart as shown here, or other organ systems that were looked at. It was just the intestine that showed exclusive upregulation of glucose uptake. So we saw that there was an increase of glucose going into the small intestine, and Alex again conducted extracellular acidification rate assays through the seahorse assay. An extracellular acidification rate is just an indicator of glycolytic activity. So he saw that in the jejunum and ileum, but not the duodenum, there was a significant elevation of extracellular acidification rate, which indicates increased glycolytic activity within the small intestine. And he then measured lactate levels within the media from these cells, and he saw that in the jejunum and ileum again, there were elevated levels of lactate, which is another end product of the glycolysis pathway. So between those two, able to see that glycolysis is functionally elevated in the jejunum and ileum, but not the duodenum. So we saw that there's lower plasma glucose, there's an increase of glucose uptake from the circulation in the intestine, and that glucose is likely being used preferentially for glycolysis. And we have bulk RNA-seq data here showing elevated glycolytic gene expression. So bulk RNA-seq, just measuring mRNA levels of the entire transcriptome for every gene. We can see that we have tissue data on the left, with each of the rows being a glycolytic gene. And on the right, we have organoids showing the same genes, with each cell being either up, down, or not significantly regulated, or with blue being the down-regulated, red being up-regulated. We can see that in the duodenum column, we have a low but still somewhat noticeable level of glycolytic upregulation with 4 genes being upregulated. But as you move progressively towards the ileum of the tissue data, we can see that there is more and larger amounts of this glycolytic upregulation that we see. And this is completely absent in the organoids, suggesting the importance of some in vivo specific signal or something intrinsic to cell culture that might be different or causing a lack of upregulation. So using that data even further, I was looking for finding and using bioinformatic tools to look at and find potential transcription factors that are likely to regulate this particular gene set. And HIF1 alpha came up to be a likely candidate. So this is the same pathway that we saw in the previous slide, but now HIF1 alpha, being a transcription factor, is known to regulate specific sets of genes. And these genes are marked in red and the arrow pointing at them. So we have in total 9 different HIF1 alpha targets through ChIP-seq datasets that are listed here in this pathway. And of those 9 genes, 25% of them are also upregulated in the duodenum, 62.5 in the jejunum, and 100% in the ileum. So all of the HIF1 alpha target genes are functionally elevated in the ileum data set, showing a progressive increase of HIF1 alpha involvement as you move from the duodenum towards the ileum. Looking at actual HIF1 alpha levels in our data, we can see that it mirrors the previous data where jejunum and ileum now show significant elevations of HIF1 alpha on the mRNA level, but no change in the duodenum. And this is important especially as HIF1 alpha is a primarily post-translationally modified protein that kind of goes through the degradation or stabilization, which we're going to talk about in the next slide. So seeing that elevated transcriptomically is also a good sign. So HIF1 alpha is known as hypoxia-inducible factor 1 alpha, and it's kind of the canonical means of degradation or stabilization is oxygen availability. So going through the pathway of degradation, if HIF1 alpha is in normoxia, it will then be hydroxylated by the propyl hydroxylases, and then ubiquinated with VHL proteins, and then degraded. So in the presence of normal oxygen, that pathway will progress as normal, but in hypoxic environments, we lose the oxygen that does not allow it to then be hydroxylated and degraded. So then it will translocate into the nucleus, binding to its cofactors, such as HIF1 beta and others, and then activating the transcription of various target genes, such as angiogenesis, glycolysis in our case, and many more. But that is only one of the ways it is able to be stabilized or degraded or regulated. Substrate availability is also a key factor. If you have lower levels of alpha-ketoglutarate, it will also prevent the degradation of this protein. And if you have high levels of succinate, it will also stabilize HIF1 alpha. So depending on what substrates you have, it can also impact that. And another last one would be oxidative stress. Iron 2+ is a cofactor of this reaction. It's a required element, and oxidative stress converts this iron 2+ to the 3+ state, which is unusable by the propyl hydroxylases. So despite HIF1 alpha being hypoxia-inducible factor 1 alpha, there are many ways that this can be regulated. I wanted to dig into that and see if it were what potentially was driving this stabilization. So here we have 4 different genes. 3 of the PHD enzymes, they're in a family of 3 different. They all perform the same task but are regulated by different means. We have the duodenum on the left, jejunum in the middle, and ileum on the right. Importantly, looking at PHD2, we can see that it is unchanged in all 3 segments, and PHD2 is the most oxygen-responsive propyl hydroxylase. Under cases of low hypoxic-mediated stabilization, PHD2 will be the primary PHD enzyme elevated, which we don't see in our data. And the second most important one would be VEGF-A. We can see that there is kind of a mixed regulation of these 3 segments. And in cases of hypoxic-mediated stabilization, this gene once again will be elevated, which we don't see. So this points towards a non-hypoxic method of stabilization. And the last one, PHD3, is elevated in any form of HIF1 alpha stabilization. So the fact that we have PHD3 points towards HIF1 alpha is being stabilized, and the lack of elevation in the other 2 genes, PHD2 and VEGF-A, point toward it being a non-hypoxic route of stabilization, such as reactive oxygen species or oxidative stress. So moving towards oxidative stress, we saw that in our bulk RNA-seq data, we have DUOX2 and DUOX-A2, and these 2 are among the top differentially expressed genes in our data set, and being the most elevated in our CF samples. DUOX2 is an enzyme responsible for catalyzing the hydrogen peroxide producing reaction, which is a major reactive oxygen species. And DUOX-A2 is just a maturation factor for DUOX-2. It promotes proper folding and transport to the membrane, or can act properly as an enzyme. And we see progressive elevation of our DUOX-2 transcripts, as well as DUOX-A2. And it is significantly elevated in all three segments, particularly the ileum, where it's a 5-fold increase compared to its wild-type counterpart. So this is consistent with elevated epithelial reactive oxygen species generation. And moving to the next slide, those were all kind of computationally driven hypotheses and predictions. And moving on to kind of experimentally validating it now, did IHC of HIF-1 alpha to look at the localization and stabilization within the intestine. And on the left, we have a wild-type mouse, and which shows nearly undetectable levels of HIF-1 alpha protein. But on the CF jejunum on the right, we can see large amounts of stabilized HIF-1 alpha protein localized to the nucleus within the villi tips. And it also extends into the crypt region as well, although not as prevalent as the villi tips, suggesting a potential bacterial involvement or something that interacts with the lumen significantly. So now we have looking at how it kind of may have RNA scope of the wild type in CF mouse conducted by Laurentier within the lab, looking at SLC2A1, which is a known glucose transporter. Hexokinase 2 in yellow is a key glycolytic enzyme. You can see in wild type kind of uniform distribution of both SLC2A1 and hexokinase 2 throughout the small intestine. And looking at our CF mouse, we can see that both SLC2A1 and hexokinase 2 have increased expression compared to the wild type counterpart on the left. And this is primarily localized again to the villi tips, as we can see here, and slightly into the crypts, but not as prevalent as in the villi tip region. So this shows similar localization patterns to our HIF-1 alpha that we see up here, with increases on the villi tip, suggesting that HIF-1 alpha may play a role in interacting and driving the transcription of some of these key glycolytic genes. So just going over a quick summary, we can see that we started with the observation that the plasma glucose was lower in our CF intestines. We saw that it was being preferentially going towards the intestinal region. We can see that there was increased glycolysis, particularly in the jejunum and ileum of the intestine. We see that glycolysis was transcriptionally elevated within our tissue while being absent in our organoids, suggesting that there is some in vivo factor, such as a microbial or immune influence. Looking at our transcriptional data, we can see that due to our PHD3 upregulation and our selective VGFA expression patterns, they point towards a non-hypoxic stabilization, which ended up being localized strongly to the villi tips. Bacterial burden is likely to drive this bacterial species or this reactive oxygen species production via the DUOX-2 and A2 enzyme. And that could promote the HIF-1 alpha stabilization, which outline metabolic adaptations within the intestinal epithelium, linking together multiple factors such as oxidative stress, microbial interactions, and transcriptional reprogramming through this HIF-1 alpha signaling. This is my overall model. CF mutations in the CFTR gene lead to either non-functional, misfolded, or non-existent CFTR protein. It's been well documented that this leads to increased bacterial load as you move towards the ileum of the small intestine. This can induce the expression and stimulate reactive oxygen species generation through DUOX-2, which would inhibit the propyl hydroxylases, stabilizing HIF-1 alpha, and elevating and starting the transcription of these glycolytic genes. Some other implications that are within CF, though. HIF-1 alpha has been shown to downregulate the production of CFTR in CACO2 cells. And while that might not play a huge role if they already have lower amounts of the CFTR protein, it may impact the effect and efficacy of modulators if there is a less lesser amount of CFTR protein for those to act on if it's actively being downregulated by HIF-1 alpha. And in CF, the NRF2 pathway, which is the main antioxidant pathway within the body, has been shown to be dysfunctional. So maybe the increase of reactive oxygen species is not able to be adequately taken care of without the body's traditional systems that it has in place. The next steps, utilize germ-free mice or antibiotic-treated mice to try to better understand specific microbial influences that may be in play and what certain species may have contributing to this phenotype that we see. It's acknowledging everyone within the Drum Lab for their support along this academic and research journey. Mark Cameron Lab, Harrison Lindley, has been helping with some single-cell analyses for some of my future steps. And the CFF for funding through various grants, and university support through the Research Institute for Children's Health, and financial support from the School of Graduate Studies at Case Western for making this travel possible. And I have poster number 339 if you want to stop down during the poster sessions to talk more about this research. And that'll be it. Questions?
So from the last group, we apologize for the online questions. They're coming through a little bit late. So if you want to try to ask a question online, definitely try to do so, but just know there is a little bit of a delay. Katie?
Katie Larsnota, University of Iowa. Question 1. Do you think this is an adaptive or a maladaptive response?
I definitely think it's adaptive. HIF-1 alpha is a big part of barrier integrity and the antimicrobial response. But I think the biggest part is that it's being left unchecked. We have a lot of reactive oxygen species being pumped out, but the NRF2 pathway that should be maybe counteracting this and keeping it in check is not adequately removing or dealing with them, and the body doesn't have enough of a fallback. And with the increased bacterial load, it might not just producing too many reactive oxygen species. But yeah, I would say it's mostly adaptive, but it can left unchecked maybe.
Okay. Question number 2. Sorry, I have 3. Question number 2 is, do you think that there is you'll see this with different degrees of CFTR dysfunction? For example, different CFTR mutations, maybe HETs? What do you think?
I haven't really looked at any heterozygote data. I think it may play a part, especially with HIF-1 alpha actively downregulating the CFTR production. If they're kind of borderline with only having the 1 mutation instead of 2, maybe this kind of pushes it even further down, which is why maybe they see some GI manifestations. I'm not entirely sure about that.
Okay, question 3. We have some human data on abnormal glucose absorption in the intestine. Are you going to look at this in any kind of human model?
It would be very nice to look at it in a human model. I think seeing how it translates to humans instead of just looking at the mice phenotypes in particular. I do plan on looking at and inquiring like human samples, biopsies, and that to see how it affects them. Yeah.
Aaliyah?
Aaliyah, which Iowa. So it's super interesting. I was just thinking like how much this is CFTR and how much is the microbiome and perhaps just a combination. So you're probably just thinking about experiments of, is this microbiome and is it CFTR specific and perhaps, getting the stool of CF and then giving it to the wild type and the vice versa and then see if you can create what you're seeing. I think perhaps just one of the experiments, because we do fecal microbiota transplant in humans, right? So I think that's perhaps one thing that you can just see because, can you create this and, glucose absorption through the intestine by doing this? So I think that's just one approach that I can think about. And the other thing, have you seen, you have shown the HIF-1 alpha and then deoxin. Have you seen actually an ROS production in the intestine of the CF mouse? And can you change that perhaps by modulating the CFTR and then is the CFTR dependent? So those are 2 questions. I don't have 3.
So I guess talking about looking at actual reactive oxygen species, since all of this was just at the mRNA level, transcriptomic data. I'm working on doing protein, like immunofluorescence on DUOX2 and A2 to see if it equates to a protein level difference. And there are some assays like DFCDA assay and other reactive oxygen species quantifications that'll see, is it being taken care of, or is it just massively elevated, or seeing how it kind of relates to the wild type levels. Because I think it would be important to see it functionally and see how it is in the actual tissue, not just transcriptomically. Thank you.
All right. If we can keep it succinct, we can probably get both these questions in, so go ahead.
Great talk. My name's Alvin Chan, Pete's GI from UCLA. I think that one of the things you showed was glycolysis is increase in the more distal elements.
Yes.
How do you reconcile that with, I think, generally these simple sugars are more taken up in the more proximal parts, like the duodenum and the jejunum. So how do you, what's the explanation for that?
I think it's being used for kind of non-traditional means, with the nerve-2 pathway being dysfunctional. And we see the increased bacterial load in the most distal parts, the jejunum and ileum. And if it's playing a role in elevating the oxidative stress in reactive oxygen species, that glycolysis may be not being used for energy, but being used for its like secondary pathways like PPP to kind of neutralize or get rid of this very oxidative environment.
Got it. So not necessarily correlating with the uptake per se, but more maybe microbiome related and glycolysis too.
Yeah, it may not be related to like where it's being absorbed, but just where it's needed most.
Cool.
Yeah.
Thank you.
Rebecca Holmankley from University of Alberta. That was really great. In the beginning of your talk, you showed really clear difference in the tissue versus organoid expression of these genes. Have you been able to go back to see if you can modulate some of these pathways that you're now kind of identifying, to see if you can recapitulate that in the organoids? And if you could, then you could kind of get at Katie's question.
Yes.
And,
I've been looking at it. There is very different expression in organoids. There's HIF3 alpha is a negative regulator of HIF1 alpha, which is elevated only in organoids. And I think there's a kind of a case for knocking that down and stabilizing HIF1 alpha through stabilizers or hypoxic stabilization. But then you have to like co-culture with microbiome. It's a lot of factors that may have to be done at the same time. Definitely possible. And it'd be something exciting to look for, but I think there's no selective HIF3 alpha inhibitors as well. So it's not the most feasible, but it'd be something that would be very interesting to try to do, to have a better organoid system to look at metabolic things.
yeah, awesome. Thank you.
All right, so just real briefly, tying off to the last two questions that were had, at least in human, and I assume it's the same in mouse, CFTR expression actually decreases as you go down the intestine, but you're seeing more of your response kind of in that distal part, going to the point of, where glycolysis occurs, where the microbiome is most impacted. The idea of teasing apart what is the driving force is going to be really interesting as you see what comment you had on that.
I think it's interesting. I think it's kind of pseudo-independent of CFTR expression. Like it's normally expressed in the crypt region, which we see the least amount of HIF1 alpha. I think it's just almost strictly maybe microbiome influenced. And then despite it being not as prevalent of CFTR expression, still having a lack of it and any downstream effects from the duodenal kind of expression, changing mucosal, and seeing how it affects the microbiome. Thing.
Excellent. As a reminder, he is a PhD candidate, and I got to say some really mature answers up here. Great job.
Thanks.
Thank you. Thank you.
All right, our next talk is going to be from Dr. Paul McNally, an associate professor of pediatrics and child health at the Royal College of Surgeons in Ireland. Thank you.
Great. Thanks very much for inviting me to speak. So I'm presenting on behalf of the Recover Study Group. Here's my NACF disclaimer and my disclosures. And my other disclosure is I'm a pediatric pulmonologist talking at a gastroenterology session. So the gaps in knowledge might become evident over time, and I apologize for that. So background and context then just to the slides that I'm going to present. So RECOVER is a multi-centre post-approval study of ETI in 8 sites in the UK and Ireland. So we look at a wide array of outcomes from pulmonary, so looking at lung structure, lung function and lung inflammation. We're looking at generic things like sweat chloride adherence, quality of life, and then a GI, nutrition, liver aspect. And I'm going to talk about the GI aspects today. So for the GI outcomes, we're looking at stool markers, so calprotectin, M2 pyruvate kinase, fecal elastase, I won't talk much about that today, and the gut microbiome. And then in terms of symptoms, we're looking at the CF abdominal symptom score, which was a specifically developed symptom score for people with CF. Okay, I'm not sure why they come up so poorly there, the pixelation of the graphs. So apologies about that. So this is the abdominal symptoms score. So we start on the left, and that's children aged 6 to 11. So abdominal symptom score on the left-hand side, study visit on the bottom there. So you'll see the numbers in blue and then the level of statistical significance compared to baseline in the stars just above each of the points. So you can see both for 6 to 11 that you're seeing on the left-hand side and also for 12 and over on the right-hand side. There's a significant drop in abdominal symptom score at the 1 month time point. It's probably maximal at about 2 months. I should say there's a 2-week recall period for this questionnaire. And then largely it's unchanged subsequent to that. You'll see on the 6 to 11, the final 24-month time point is a very small number, so I'm not sure we can read too much into that for now. So to date, we've seen an improvement in abdominal symptoms independent of age in this reasonably sizable cohort. When we look then at inflammation and fecal elastase over time, this is 6-month data. So you can see in the light, I don't know what you'd call that, green or blue, depending on your preference. In the lighter colours is the children aged 6 to 11, and in the darker colours, those 12 and over. So if we start at the left-hand side, looking at calprotectin, there's a couple of things you can see. Number one, a very wide spread in the data, and again, that's not that surprising. So despite that, in the darker bars there in the older group, you can see moving from above the upper limit of normal down to the lower limit of, below the lower limit of normal, or below the upper limit of normal in the older group, and that was very significant. More normal values in the younger age group, so we were able to see a statistically significant difference at 1 month, but that wasn't sustained at 6 months, and that's probably a numbers thing, and I'll show another type of analysis in the subsequent slide. Second of all, in the middle section there, you can see the percentage of individuals with an abnormal M2PK. So M2PK, as most of you will probably know, it can be used as a marker for intestinal cancers based on its measurement of cell turnover. And in the older age group, very abnormal, or a high proportion of people with an abnormal level, and then statistically significant reduction over 6 months. Less abnormal in the younger age group. And then finally on the right-hand side, the fecal elastase, again, a lot of 0 values here. So in this older age group, it's not really that meaningful, other than the fact that the levels are slightly higher in the younger kids, and that's no big surprise. But we certainly want younger data and more longitudinal to read into this to any great degree. And then just another way of analysing the stool calprotectin levels, and this was Luke Hoffman's group, the PROMISE GI cohort. This is very much modelled on their analysis, where they looked at whether you were normal, abnormal or borderline, and you can see that both in the younger age group and the older age group, there was a significant change in the proportion of people who were in the normal, borderline and abnormal groups. And so this is a much more meaningful analysis, I think, than the previous analysis that we published. Okay, so We've seen to date improvement in abdominal symptoms, improvement in gut inflammation and markers of cell turnover. So now we wanted to look at the microbiome. And what we were really interested in when we started the program was to look at the relationships really between inflammation, symptoms and dysbiosis to see could we pick out any static relationships or any relationships in the changes over time, these variables on ETI. So this is an interim analysis. We've had, because this is a multi-center study and we're shipping samples all over the place, we've had a lot of issues with cross-border shipping. So anyway, we don't have all of our samples at home and all analysed yet. So you can see that we have these 4 time points, baseline, 1 month, 6 months and 24 months. And for the 24-month values, we really only have children aged 6 to 11 as opposed to 12 and over. So I'm going to show you some analysis in the younger age group up to 24 months and then in the older age group only up to 6 months. Okay, so starting on the left, this is Shannon diversity. And you can see in our cohort in the 12 and over age group, we saw a marginal change at 1 month, but really no change at 6 months. And so the PROMISE group, with a much larger cohort in what they published just in the 12 and overs, did show a statistically significant difference that was only evident at 6 months as opposed to 1 month. So we didn't quite see this, and this may be a numbers issue. In terms of richness then, again, no significant difference over 6 months. When we look at the 6 to 11 age group then up to 24 months, and again, smaller numbers here, we're not really seeing any change in Shannon diversity, which is on the left-hand side, or species richness on the right-hand side. You can see, obviously, there's a big spread in data here and it makes it harder with smaller numbers. We then, for the 2 age groups, 6 to 11 on the left and 12 and over on the right, did a PCOA analysis just to look at overall community composition and see if there was a shift with treatment. So on the left, you can see the bold dots in the centre and the spheroids, that we really didn't see any meaningful change in community composition. And then same in the 12 and over age group, reasonably tight over a 6 month period. So nothing statistically significant here in terms of change of population. But then similar to Luke's group, we wanted to look at, not looking at time on treatment here, but just looking at fecal calprotectin levels and segregating the whole group in 6 to 11 and 12 and over into normal, borderline and abnormal fecal calprotectin. So the PROMISE group did show a significant, that the normal levels of calprotectin, they had a very different microbiome than anybody with borderline or abnormal, but we weren't able to demonstrate that in our data, although there was a bit of a hint to it in the younger age group. We then want to look at our younger versus our older age group. So on the left-hand side is diversity, and this is all time points, 6 to 11 versus 12 and over. And we were expecting a difference here, but didn't really see it. But we did see a difference in richness. There was a higher richness in the 12 and over age group than there was in the 6 to 11 age group. But again, you can see the amount of spread in the data there and how difficult it is to find any statistically significant differences. This is a random forest analysis on our data. So it basically takes the top 20 species that are associated with gut inflammation measured by calprotectin and ranks the importance of these in terms of community composition and relationship to infection. So some of you guys might know these names, but they're certainly not household names to me. So in the 6 to 11 age group, what we saw was very different to what we saw in the 12 and over age group. Fickia cola and Rosaburia were the 2 species that were most associated with inflammation in the younger age group, albeit inflammation was much more limited. And then when we look at the older age group, there was a really dramatic standout here, which was Staph aureus, which was really mostly associated with inflammation in the adolescent and adult age group. And this is not dissimilar to what the PROMISE group found. But for us, this was the only organism really that seemed to be heavily associated with inflammation in the older age group. So the final thing we want to do then is look at all of these things together. So I'll just orientate you. It's a little bit hard to get your head around. So these are correlation heat maps looking at the relationship between M2PK, calprotectin, CF abdominal scores, and then diversity and richness in the microbiome. So top left hand is that's a static correlation at baseline. Top right is a static correlation at 6 months. And then the bottom one is a correlation in the changes over that 6 month period. So what we see at baseline is there's really, except for the relationship between M2PK and calprotectin, which is kind of expected, and the relationship between richness and diversity, there's no other strong relationships between any of these endpoints. But what we do notice is at 6 months, the heat map looks very different. So the relationships have changed within 6 months. So the relationship between calprotectin and M2PK has gone. And again, that's not a surprise because they've both improved. But the relationship between richness and diversity has stayed, which is interesting in itself, perhaps not expected because there wasn't a huge shift in the microbiome based on what we saw. And then when we look at the change over time, again, because of the reduction in the inflammatory variables, not a surprise that the change of M2PK and calprotectin were associated, but there was an association between change in richness and change in diversity, which is a little bit unexpected based on the data that we've shown. So over 24 months of ETI treatment, we saw sustained improvements in symptoms of gut inflammation. Over 6 months of therapy in our cohort, so far we haven't shown much in the way of change of microbial community composition, but we will have bigger numbers and longer time points in due course. And we haven't seen a significant relationship between symptoms, inflammation, and microbiome, apart from things that are inherently related. So, and we saw very different profiles in that random forest between the younger and older age group, and that's something we're going to look into in greater detail. So we're currently completing the shipping and analysis of all of our studies, our samples from the parent study up to 24 months, and we really want to do some deep dive on species level analysis. Recover is in an extension at the moment. It's 2 years for parent study and then another 5 years of extension. So we'll have 7 years of data in due course. And our the next way we're moving with this is to the younger age group, because we're very interested, rather than taking people with established dysbiosis and following them, looking at kids from birth with a control cohort of healthy comparators. So ENHANCE is looking at that initially over 5 years, but hopefully up to adulthood, and that's annual sampling. And then the BUNNY study is looking at very frequent sampling in that 0 to 2 age group, where there's a lot of changes in the microbial community, and comparing that to controls. So I just want to thank all the participants and everybody involved in the Recover study, and particularly Rachel Cregan, our project manager, who just ties everything together for us. Thank you.
We'll start with Aaliyah.
Okay. Aaliyah, which University of Iowa. So very interesting study. Can you clarify a point? So especially about the samples for patients over 12 just being so low. Is that just the study not completed yet, or just the samples not collected or not yet collected?
Yeah, so the samples are collected, but they're sitting in sites, and they haven't all been shipped to us. And then some of the samples for calprotectin and M2PK were analysed in Germany, and we need them to ship the samples back to us to ship them to our microbiome lab, and we've had a lot of cross-border stuff. So all the samples are there, we just haven't got them shipped and analysed yet.
Okay, that's helpful. Thank you.
Mike Narcoitz, Colorado. The abdominal complaints are different than what we've seen in PROMISE. You use a different scoring system. But a comment and a question. The average score at the outset was 15. The CF abdominal score goes up to 100. So my interpretation of that is these are relatively mild symptoms. So have you done an analysis looking at a cut point for the CF abdomen where perhaps you enrich it in individuals with more symptoms and try to compare that to those with no symptoms using CF? So if you use a cut point of 40 or something, and I don't know what the right number is, but you might think about that, because we've done that in PROMISE, and appears to be a little bit more of a difference when you look at those with more significant symptoms. And I just think you have relatively not much, and they're real to the patients, but they're, from how they score them, it's pretty low.
Yeah, that's a great question. We haven't done that analysis, but that's a really good point. Thanks for that.
Hi, Hillary Hayden from UW. Appreciated the talk and enjoyed seeing the random forest data. Wondering if you looked at directionality using a mixed effects model. So you showed us the top associated taxa, but we don't have a sense of whether those went up with ETI or down. Were you able to look at that? Are you planning to?
We did some of that, but it's kind of hampered by the numbers. The big one we looked at was staff. I didn't show that data, but we didn't see a significant difference in staff in the older group on ETI. There was a I'm trying to remember the name, Wexelleria or something was the only species that we saw in the older age group that significantly changed, and it went up as opposed to down. And it's been reported elsewhere to be associated with protective effect. So we didn't see much in terms of change. We did in the younger age group, but the ones that changed were not the top two that were associated with inflammation in the random forest. But that species level analysis, we're going to be able to do much more of that when we get all the data in and up to 24 months.
Yeah, great, thank you.
Hi, great talk. Eliana from Case Western. I was wondering if any of the species that you had the high level burden on, if you guys did any plating with it to see what the logarithmic burden of the bacteria was.
No, this was all. We didn't do any culture or plating of any of the samples. These are all literally, taken, aliquoted, and sent to the different places. So they.
It was all sequencing data.
It's all, yeah.
I think it'd be really interesting if we could look at the actual burden of the bacteria and correlate that with the inflammation. It's a little bit.
Yeah, I'm not sure. I'm not a microbiome expert. This was all done by collaborators. They did metagenomic analysis, so I'm not sure is there sufficient in that. This wasn't 16S or whatever. This was kind of pretty high-end analysis, so they may have some of that data, but I just don't know.
Yeah. Okay, thank you. And then I also was wondering if you guys had any data on the antibiotics that patients may or may not have been on.
We do have that data. It's a little bit delayed. We have IV antibiotic data from exacerbations, and we have collected in some individuals oral antibiotic data because we're doing a big adherence arm to the study. But it's very delayed coming in because we've got to retrospectively get pharmacy reports and that. So we don't have all of the oral antibiotic data for these cohort. But there was a significant decrease in, obviously, IV and oral antibiotic use. And so that's a bit of a confounder as well.
Yeah. I mean, it'd be interesting to know what antibiotics is that would drive some of the species that are being.
Yeah, although we don't have, we don't have all the years of retrospective stuff, which may be more influencing it rather than short-term stuff.
Yeah, for sure. Thank you.
Time for one more?
Yep. Yeah.
Hi, Pavan Bhattacharjee from University of Washington. So I think you've shown nicely that inflammation goes down, but the microbiome doesn't change. So can you speculate why you think inflammation is going down?
Well, I think, again, look, this is like a squirrel playing Jeopardy. I don't really, I'm just totally guesswork here. But I think from what we're seeing in the lung microbiome, I think there's a kind of an inflection point that you get to a certain point, you're not really going to change what's been there for a long time. And so I think some things are slower to change and some things are more rapid. I think like in the lung, you see a more rapid change in inflammation than you do in microbial community. And there's different things impacting. There's epithelial stuff and then there's microbial interaction stuff. So I don't know, it's too early for us to know, but I think the longer we go on with this data collection and the more we have all the different endpoints coming together, and we're looking at sweat and we're looking at lung inflammation and we're looking at all those other factors in a mixed effects model, it might help us to tease it out a little bit.
Right. Yep. Dr. Josie from Charlotte, I just wanted to find out, did you control the diet? Because I know maybe the diet over there is different from here, and that seems to affect microbiomes as well. And the diet in the 6 to 11 and adolescents are usually different. I just want to know if there's any control for that.
No, we certainly didn't control for it. Neither did we measure it. We had to cut ourselves. We had to stop collecting data because the study was just ballooning and we were getting too many things, and it was just unmanageable. So actually, we had to just pick our battles, if you like, and that's data that we don't have, unfortunately.
All right, we have time for one more.
Yeah, one final thing that may affect the microbiome composition is we looked at transit, orocecal transit, and the changes from around 6 hours pre-ETI to 4 hours once you've been on ETI for a good length of time, but you need to be taking it for about 18 months. So that more rapid transit is likely to have an effect on the microbiome content as well, and symptoms.
Yeah, unfortunately, thanks, Alan. Unfortunately, we don't have that data either. It would be lovely to have all of this together.
Gotcha. Excellent. Just one final comment here, piggyback on what Mike said. I want to give everybody else enough time to talk. So, disclaimer, I was a PI in the Galaxy study that set the PROs, but if we look at the PROMIS in our data, it was about 30% of the population had severe symptoms on our scale, and those were the ones that had the response to ETI. If you look at everybody that had moderate to mild, there was no symptoms, so just kind of piggybacking on what Mike said,
great.
looking
That's really
at the
useful.
more severe symptoms.
Yeah, really useful. Thank you.
All right. Thank you.
Thanks.
All right, guys. Great conversations. Keep them coming. Next, we have Omar Morales, who is a PhD candidate at University of Saskatchewan in Campanucci's lab, and he's going to be talking to us about a gut on a chip.
Okay, hi everyone. Thank you for coming here, and thank you for the organizers for inviting me to give this talk entitled Gut on a Chip Model to Study the Gut-Enteric Sensory Neuron Interface in Cystic Fibrosis. There's no relationships to disclose related to this presentation. So GI complications in cystic fibrosis have become super common, and recently those complications are being investigated. These complications include, but are not limited to, stomach cramps, heartburn, bloating, lack of appetite, nausea, wind or gas, loose motions, and vomiting. Some GI luminal complications are being considered secondary to the decrease in CFTR function. This decrease could lead to the small intestine bacterial overgrowth, good hormones dysregulation, dysbiosis leading to inflammation, fat malabsorption, and pancreatic insufficiency. Also, this decrease in CFTR function has been reported that has an alteration in intestinal mesenteric ganglia and also in the enteric nervous system. These alterations can lead to an abnormal iliac smooth muscle function. So the motility and secretions are controlled by the enteric nervous system, but modulated by the feedback of sensory neurons. So we are really interested in studying the gut-brain axis because preliminary data from our lab shows that sensory neurons coming from a swine model of CF are hypoexcitable. This after being stimulated with capsaicin. Here we can see that the response of the CF neurons is not as strong as it should be as a counterpart in wild type. Also, it's been reported that they have a disrupted chloride homeostasis. And additional to this, the CFTR pathway has been downregulated. So to study this gut-brain axis, first we isolated intestinal organoids and the enteric nervous system from different models. Here these are general methods that we use. So first we isolated the proximal portion of the jejunum, where we isolate the crypts, and we seed it into matrigel to allow the intestinal organoids to form. At the same time, from the same portion of the jejunum, we coated longitudinal, and we isolated or separate the mesenteric plexus. This mesenteric plexus was then mensed and then seeded into matrigel domes to allow the enteric neurons to develop. So here I show you how these cultures forms over time. So first, in the intestinal organoids at day 0, here we have our crypts. And after 2 days, we have this spherical organoid. And after 9 days, we can see that they have these growths or bumps on the organoid. These bumps are equivalent to the crypts in the intestinal native tissue. After 15 days, we can see that the organoid grows and it's more complex. For the ENS, first we have here in day 0 our enteric plexus, and after 4 days we can see that the neurons start crawling out of the enteric plexus. After leaving the cultures for around a month, we can see that after 24 days we have a huge population of different cells, and after 28 days we saw the formation of these ridges. These ridges are under the microscope, we can see that they are contracting, so assuming that we have a smooth muscle in all culture. So for the characterization of these cultures, first we started with the intestinal organoids. We use SO1, which is a marker for epithelial cells, and also secretory cells that we use MUC2. For the ENS, we use DAPI for nucleus and tubulin beta-3 to mark the enteric neurons. And we can see how the neurons start crawling out of this explant of tissue. When we evaluate the expression of CFTR in the intestinal organoids, we saw a few cells that had a higher concentration or expression of CFTR facing the lumen of the intestinal organoid. This is really interesting to us because it can suggest that they have a different kind of cell that could be addressed in the intestine to future or possible therapeutics. So here in this video, I show you how these ridges start contracting, and then with the red arrows, I show you how the neurons wrap or are surrounding these ridges. So for the characterization of this, we use different markers. Again, we use tubulin beta-3 for neurons and actin to see the structure of the neuron, and smoothilin to label the smooth muscle. So after 15 days in culture, we can see how the enteric neurons start to surrounding these ridges, and we can see a population of smooth muscle in the middle. After 20 days, we can see that there we have a more defined structure and also more integrated neurons in it. And after 28 days, we can see a really well-defined structure and a complete integration of the enteric neurons. So now that we have most of the components of the gut-brain axis, for example, sensory neurons represented in orange in the cartoon, and this is a microphotograph from my cultures. We have the enteric neurons represented in green. This is another microphotograph of my cultures. We also have the intestinal smooth muscle, and we have the organoids which represent the epithelial from the intestine. So we start working in the gut-on-a-chip model because we're interested to see which component of this gut-brain axis could be more affected by the lack of function of CFTR. So for this, we start first with direct co-culture, which is where we isolated sensory neurons coming from DRGs, isolated from newborn pups. So the enteric neurons were isolated from the enteric plexus coming from adult animals. These two types of neurons were sitting along the intestinal organoids into matricial domes and to evaluate how they interact. So in these videos, I show you how the sensory neurons projected processes through the intestinal organoids, and we can see that they have several points of contact in the surface of the intestinal organoid. The same happened with enteric neurons, where we can see that the enteric neurons project these processes directly into the intestinal organoids, and we have also different or several points of contact in the surface of this organoid. So to evaluate the behavior of the sensory neurons, one of our students performed patch clamp electrophysiology. So the goal of this is to see if the neuron behaves as the conventional culture of sensory neurons, and we can see that the sensory neurons can fire several action potentials. Also, we're working in microinjections of bacterial proteins relevant for CF. Right now, we're working with LPS. So the goal of these microinjections is to see how the sensory neurons or the enteric neurons responds to the stimuli of different proteins loaded inside of the organoid. So here I show you how we use a microneedle loaded with LPS along with a fluorescent tracker, just to see that we are in the lumen of the organoid, like in this image here, and we are not stimulating directly the sensory or enteric neurons. So one of the questions that we had before, it was that the neurons are directing their processes to the organoids, or it was just chance. So for this, we use a microfluidic chamber. In this microfluidic chamber, we have 3 compartments, in where one of the compartments we see it with sensory neurons, the second one we see it with enteric neurons, and the third one with intestinal organoids. These compartments are separated by a barrier, but also are connected through microgroups. This is a microphotograph of this microfluidic chamber. We have the 2 compartments separated by the barrier and connected to the microgroups. These microgroups has a diameter of 10 microns, which allows the processes to cross from one compartment to another, but not the cellular body, because this is bigger than the diameter of these groups. So when we see these two different types of neurons and the intestinal organoids, we can see how the sensory neurons projected their processes from one compartment to another one, and we can track how these processes are directed in this microfluidic chamber, here represented with this red dashed line. So we can see that almost reaches the third compartment where the intestinal organoid is, right here. So the same happened with the enteric neurons, where we can see that the processes are being directed to this compartment, and one of those, it was almost reaching it. So this suggests that the organoid secrete chemioattractants to the neurons to be innervated by them. So the goal of this gut-on-a-chip system is to use it as a model in CF. So for this, right now we're working with humanized models, where we have a mice model with humanized CFTR without the mutation, and also we have the CFTR with the mutation F508 elite. From these models, we can isolate the sensory neurons, enteric neurons, and intestinal organoids to evaluate how they behave. So right now, we're working with the alteration of those regions that I mentioned before. And we can see that in the wild type, in the control group, we have this rhythmic and constant pattern of contraction. Meanwhile, in the CF, we have some contractions, and then we have something flickery right there. Unfortunately, fortunately, actually, after the treatment of ETI, we can see that the CF model recovers this beating or this pattern being more smooth and constant. But after analyzing these videos, we saw that yes, in fact, the frequency is restored, but the amplitude of this contraction decreases. So this might give us an insight why people with cystic fibrosis don't show any improvement after being under the ETI treatment. So for further steps, we're going to test these different CFTR modulators in intestinal organoids and also in the interface of intestinal organoids and neurons interface. In addition, the chamber will be keeping using for further characterization of the epithelial cells and neurons. And finally, we're going to perform calcium imaging in the ridges of the muscle to elucidate the dynamics of the intestinal muscle and the neuron innervation. So for conclusions, we developed a gut-on-a-chip system to study the gut-neuron interface in cystic fibrosis, which could provide valuable insight into the effect of inflammatory signals, microbiome, and CFTR modulators in cystic fibrosis. Also, this system conserves the architecture of the gut epithelia, together with the extrinsic innervation of the different type of neurons. And also, this allows us to perform heterogenic cultures, meaning that we can combine CF neurons with wild-type organoids or vice versa, which would be really interesting to see. And finally, organoids provide a more accurate model for testing new drugs and potentially speeding up the development of effective treatments. I want to thank my supervisor, Veronica Campanucci, along with all my lab members for all their help and support, and of course the funding agencies, and a special thanks to the Mexican government for funding my studies. Thank you.
All right, I'm going to start with 2 questions while other people get up. Have you considered the potential release of the incretin hormones from the organoids and the role that could play?
Yes, actually, we're planning to, actually, we're doing it right now. We're collecting the media and we're going to send it to analyze for metabolites and see which hormones or which metabolites are present. And from there, we'll start making a screening to see which one is the more involved and testing to the, into the neurons to see how they behave.
Cool. And then one of the components that isn't in your amazing model is the lymph that gets is throughout the lamina propria, and of course is where dietary fat is secreted into. Have you considered either adding lymph to the organoid milieu or approximating nutrient absorption on that side?
We haven't considered it, but it's a really good observation. The good thing about this model is it's so flexible. So we can add lymph, also we can load the organoids with microbiome coming from CF models or wild type. And yeah, it's really, really flexible and it's really good observation.
It's really impressive.
Thank you.
Yeah, thanks. Okay, go ahead.
Hi, really nice talk. My name's Danica from Case Western. My question was about the profile of the neurons that you're using in your culture. So you've shown really nicely that they grow and make contact with your organoids. Do you know that functionally these neurons are similar to the neurons that you would find that innervate the gut in vivo? So do they make all of the same receptors and secrete the same neurotransmitters to validate that those kinds of interactions are what you would see in a gut that accepts food and has the like motor neurons involved in contractility and that kind of thing?
Yeah, actually, we're working in the characterization of this model, including all the things that you're saying, what kind of neurons we have, which neurotransmitter they're secreting, what receptors they're expressing. Yeah, really interesting, but we are working on that right now.
Okay, awesome.
Thank you.
Thanks.
Hello.
Great study. So I have very basic questions. When you co-culture neurons and organoids together in metroderms, how do you control the media? Because these are two different media.
Yeah, we noticed that the organoids apparently are more sensitive than the neurons, which for us was kind of odd because usually the neurons are more picky, the way to speak. But the only thing that we do is enrich the organoid media with NGF, which is a factor for growth in neurons. And they are really, really happy. And also that was the other goal of my student to perform patch clamp to see how they behave with that media. And they like it. Yeah.
So it is NGF media?
NGF.
Okay.
NGF is a factor. It's a neuronal development factor or something like that. But yes, NGF.
Okay, thank you.
Thank you.
Amazing. Another fantastic PhD candidate talk. All right. For our last talk of the session, we're going to hear from Dr. Rosera Bass, an assistant professor of pediatrics at Nationwide Children's Hospital. Welcome.
Stay on time. Wait, I don't even have to look at that. Okay. I was going to try really hard not, when you're the last one before lunch, the pressure is really on not to go over. But thank you for everybody also finishing on time so I don't, a little less pressure off. So I'm going to swing this really in the other direction. We've seen a lot of basic science, and this is going to be primarily a clinical research talk, looking at a novel tool for the assessment of gastric motility in youth with CF. And I'm Rosera Bass. I'm a pediatric gastroenterologist and nutrition specialist at Nationwide Children's Hospital. I have no disclosures, but this work was supported. These are the first preliminary data from my CF Schwachman Award, and much more to come. The data that I'm going to be showing today are from the Children's Hospital of Philadelphia. I then moved, and my Schwachman moved with me, and this work is ongoing. And so I would just like to acknowledge all of my collaborators at CHOP, Nationwide Children's, and the CF Foundation for really making it possible for me to continue this work. I was going to highlight that Jay was one of the PIs of the Galaxy study, but he already did that. But this is, I think, a very helpful graphic that I pulled from the Galaxy study that really highlights the overall symptom burden of GI symptoms in people with cystic fibrosis using a patient-reported outcome measure that Jay and Meg and everyone found really captures GI symptoms in people with cystic fibrosis. And I think it's important to acknowledge that in adults with CF, over 20% of people are reporting upper GI symptoms, stomach fullness, bloating, stomach or belly visibly larger. So certainly very prominent in people with CF. And I think that what I've really taken away from this session, and I hope we would all acknowledge, is that GI symptoms do not mean a diagnosis. And people with cystic fibrosis, as we've seen today, really have multiple different abnormalities and opportunities for mucosal disease and inflammation from top to bottom of the luminal GI tract, as well as the hepatopancreatic biliary tree. But I think that the one thing that we haven't talked much about this morning are really disorders of gut-brain interaction, the artists formerly known as functional abdominal pain. And so these are GI symptoms that can occur in the absence of structural or biochemical alterations. They may be related to motility or increased visceral hypersensitivity in the GI tract. And I think that the kind of first statement and that general definition of disorders of gut-brain interaction can be a little challenging in disorders like cystic fibrosis, inflammatory bowel disease, chronic pancreatitis, in that hypersensitivity can coexist in the presence of organic disease. And I think that it's very important to acknowledge that, and that while these symptoms may be present and underlying disease may be present, some of these symptoms may be amplified by disorders of gut-brain interaction. And so I think that these can't be mutually exclusive. Disorders of gut-brain interaction are common in both children and adults with CF. In a study of youth compared to matched controls, 36% of youth with CF versus 10% of controls met criteria for at least one disorder of gut-brain interaction. And Hay et al. In the UK found that 65% of adults with CF met the Rome-4 criteria for irritable bowel syndrome. So are DGBIs caused by abnormal motility? And I think that these words sort of get tossed around interchangeably, but I think it's important to acknowledge that motility is really only one part of the DGBI problem, and for some people may not be a part at all. And so when we think about disorders of gut-brain interaction, we think about sensitizing medical events, and we've certainly seen many reasons today that people with CF can have sensitizing medical events or reasons for GI symptoms, but also sensitizing psychosocial events that really impact how pain is perceived. And if you look at the bottom of this chart, I think we can all really imagine that many of our patients with CF are also a setup for these sensitizing psychosocial events. All of this leads to changes in the way that pain is processed, both by the nerves and the brain, and then ends up with abdominal pain or other GI problems. And so I think that while we often use the term motility disorder, and in some patients, dysmotility may be a driving force, I think it's important to consider it in this broader context. And also to acknowledge that some of these psychosocial events may be even more predominant players than motility. So how do we assess GI motility in the general population and in people with CF? So we have non-invasive tools like a gastric emptying scan or breath test, which really lacks a lot of sensitivity and specificity and is not able to correlate with real-time symptoms. We have invasive tools like AD manometry, which is a sedated procedure with catheter placement, pH impedance, which is a 24-hour study. I actually did a pH impedance study in 10 children with CF, and I can pretty much guarantee that none of them would do it again unless I was paying them. There's empiric trials of pharmacotherapy, and this is really, I think, what happens most often is we will often try in patients with kind of non-specific GI symptoms, we'll use medications that target GI motility. But what if motility, again, is not the driving force, and how can we better assess this? And so this leads me to body surface gastric mapping, which is a tool that I'm using in my research as part of my Schwachman Award. So body surface gastric mapping is a non-invasive tool. It's a 64 electrode adhesive array that operates very similarly to the way that an EKG lead would operate in measuring the myoelectric activity of the heart. It measures from the cutaneous surface depolarization and repolarization of the gastric muscles, and it correlates with real-time symptoms over the course of the study. There are normative values. It's FDA approved in adults. Pediatric normative values are on their way in the coming months. And one thing that I think is really exciting is I feel like sometimes in CF, we wait till things have been tested and figure it out in the general population and then apply them to CF. And it's been really cool to be able to sort of use this in real time as the device is being developed in the general population. But they are still a few steps ahead of us, and it's really great to have their normative data for comparison. So the real purpose of this is just to look at the feasibility of body surface gastric mapping in people with CF. And then this is just the underside of the array. You can see those adhesive electrodes. And then this is the measurement of the gastric slow wave propagation. And then what else happens during a gastric mapping test? So the array is placed. There's a 30-minute fasting period. There's consumption of a test meal, which is a Cliff Bar and an Ensure. And then there's 4 hours of postprandial recording. I generally tell people when they're doing this test that the number one side effect is boredom because you have to sit still for 4 hours. But really, other than that, it's not that bad. You just sit. And every 10 minutes, you are prompted by a participant-facing app that asks you about 6 different GI symptoms. And if you're having symptoms in between those 10-minute intervals, you can add that in. And so this is the spectral analysis that we get from the body surface gastric mapping device. And I'll break this down. So the first thing is up here on the, I can see the mouse. So up here is the principal gastric frequency. So a normal gastric frequency, regardless of meal response, is 3 CPMs. And you can see that this is like a really nice tight yellow band around that 3 CPM mark showing normal gastric frequency. And this device also can allow us to see sort of how tight that band is and how stable the rhythm is with the gastricolimetry rhythm index. And I'm going to show you later an example of a very abnormal one. And then also it calculates gastric amplitude, or the amplitude of these slow wave contractions. There are normative values, like I said, in adults, and also in pediatrics, we see a similar bump in gastric amplitude in response to a meal that then eventually comes back down to normal. And again, concurrently, we have BSGM symptom analysis. And so those every 10-minute symptoms that are recorded throughout the study are mapped on the same x-axis, the same time axis as our spectral analysis. So we can really look and see what symptoms are happening at the same time as spectral changes or no changes. And so these are 2 examples of BSGM symptom phenotypes. The profile on the right, is the continuous profile. And you can see, I'll just tell you that that amplitude is normal. And this person is reporting an 8 out of 10 upper gut pain throughout the duration of the study. And so what this is suggesting is that this person is certainly having pain, but that pain is probably not being caused by abnormal gastric slow wave propagation. In the person on the right, this is referred to as a sensory motor profile. And you can see that there is a bump in amplitude that is correlating with a bump in GI symptoms. And so in that person, it may be more suggestive of a symptom coming from a disorder of gastric motility. So this was, again, a pilot and feasibility study to examine the feasibility of using this device in youth with CF. And then because we didn't have normative values at the time of this study, we compared them to a matched healthy control population from the Auckland pediatric study. Our hypothesis was this would be feasible using our standard protocol. We recruited youth with CF with exocrine pancreatic insufficiency. They had all been on Trikafta for at least 1 year and were between 7 and 25 years of age. And then these are the exclusion criteria for use of the device itself: pregnancy, adhesive allergy, and open abdominal wound. And our controls came from 3 different centers in the US, New Zealand, and Australia, and actually many of the US controls came from CHOP as well. So we use the same study methods. The only difference in pediatrics is if children are under the age of 12, we do 2/3 of the standard test meal. And here's some of our demographics. So overall, we had similarly age and sex matched CF and control cohorts. Our participants with CF had overall normal BMI. Their median Z-score was slightly less than 0, which is the 50th percentile. They were all white and non-Hispanic. We had successful completion of the study in all of these subjects and successful spectral tracings in all except for 2 who were excluded due to excessive artifact. Overall, I think, especially from our discussion today, I think it's very important to acknowledge that this was not a high baseline symptom cohort. So again, looking at that PAGI SIM score, which is a Likert scale of 0 to 5, the median or the mean PAGI SIM scores for each of these symptoms is generally between 0 and 1, with just 2 being a stomach fullness of 1.23 and unable to finish a meal at 1.15. So overall, a low symptom burden cohort for our first test of this device. And so what did we see? So interestingly, we did see that our pre-meal gastric amplitude was lower in CF than our healthy control population and likely drove an overall lower value in the when we looked at the full study. And then our principal gastric frequency generally was pretty similar between the two cohorts. Only at the 0 to 1 hour post-meal time point did we see a significant difference between CF and controls. But what I think is really, you can sort of see on this graph, and I'll show you on the next spectrogram, there's a lot of spread in the CF, the blue CF, and you can see that the principal gastric frequency, which is really supposed to be in adults between, and similarly what we've seen in peds, in like the 3.3 to 2.6 is like a normal range. And we're seeing people with CF with gastric frequencies that are much higher and much lower than that. And so here's an example of actually one of our spectrograms from one of the people in our study with CF. And you can see on the top, I'm showing you again normal, which is that really tight yellow band across the middle of the spectrogram, indicating a 3 CPM frequency that's very stable across the study. And on the bottom is our participant with CF. And you can see that that spectral tracing is really all over the place. And so there's really wide variation in the frequency of gastric contractions throughout the course of the study. And this is expressed in that gastric allometry rhythm index, with the lower limit of normal for adults being 0.25, and in this subject was 0.14. And interestingly, in this same subject, this was one of our few subjects that actually did record symptoms during the study and had a pretty consistent symptom burden. Well, not, it was, I think, in like the 4 range, but still pretty consistent symptoms over the course of the study. And so I think this is the type of person who I'm really interested in learning more about. And then finally, looking at the rhythm index, again, in our aggregate of population with youth with CF with generally low symptoms at baseline, low symptom burden at baseline, there was only at the 1 to 2 hour we saw a slight difference between CF and healthy controls. But overall, we learned that this is a feasible test in people with CF. 86% of the participants in our study had interpretable results. And since I did this, we've actually done 7 more tests at Nationwide, and all of those have had interpretable results. So in a population of youth with CF and minimal GI symptom burden at baseline, we're seeing some small intrinsic differences in motility compared to controls. And then at the individual level, we're maybe seeing some signal that there are some motility abnormalities in people with CF. And I think the real important question, and the question that will help us to understand whether this test is worthwhile, is to know whether these differences impact clinical symptoms. And so certainly that is our next step. So we're working on completing our data collection at Nationwide Children's Hospital. We'll get some preliminary data to inform sample size and power analysis. And then for our next pilot, we're really going to be focusing on recruiting people with CF with active GI symptoms, looking at short-term repeatability of this measure, and also doing some qualitative interviews to assess tolerability of this measure and if this is something with that people with CF would actually do in the clinical setting. So I'd like to extend a huge thank you to all of my collaborators at CHOP and the University of Auckland who helped with this kind of preliminary part of the study. And I'd also like to acknowledge all of my new collaborators at Nationwide Children's who have really helped to move this study and make this ongoing work possible. So thank you so much.
I'll start with a question. Really nice talk. Are there treatments for changes in gastric motility?
So there are. There are treatments for changes in gastric motility. There are medications that can sort of amplify contractions, and then there are also medications that increase gastric accommodation. And I feel like those are often the go-tos. And sometimes they work and sometimes they don't. And interestingly, in adults with DGBIs, they've been able to use BSGM to predict response to prokinetic therapies.
For gastric mapping, how do these things compare to like a gastric emptying study or the intraduodenal manometries?
Yeah, that's a great question. So gastric emptying, the advantage of this is that you have this real-time symptom recording. So gastric emptying, you do the study once, but you may not have symptoms on the day of the test. You may have symptoms on the day of the test, and we often see people with normal emptying and lots of symptoms and vice versa, especially in CF. AD manometry is definitely our gold standard test, but it's highly invasive. It requires sedation. You have to be at a motility center, and they've been able to show that BSGM has good correlation with AD manometry. So it might be an alternative.
Thanks.
Hillary.
Go ahead, Hillary.
Hi, Rosera. Thanks so much. That was great. Hillary Bashaw, PEDS at Dornbecker in Portland. I'm wondering with my nutrition hat, how did PERT dosing compare for the different patients with their test meal and what was their range of dosing?
That is a great question, and I did not look at that, but I'm going to go back and
Future
do it.
topic,
Thank you
yes.
very much.
Thinking about what might affect symptoms and if
Yes,
they're underdosed.
Absolutely. Thank you.
PERT dosing, pancreatic enzyme replacement therapy dosing. So is their dose matching their meal? Could their symptoms be from underdosing of pancreatic enzyme replacement therapy?
Yeah, that's great. And one other piece of data that we did collect as part of this that I'm not showing here is we actually have a lot of dietary recall data to go along with these subjects. So I will be very interested kind of in our future analysis to look at how dietary intake sort of impacts this overall milieu.
Hi, thanks for the talk. Danica again. From
Let's go here. Let's go back over.
Sorry.
No worries.
Oh.
Okay, so Ian Stewart from Imperial. So fascinating talk. I was wondering whether you just mentioned that the BSGM could predict treatment response. So in other diseases beyond CF, have they looked at this as a clinical endpoint?
Yes.
As a sort of objective clinical endpoint?
Yeah, so this is new. There's, I think, still only at 4 centers in the United States, but I do know of at least one person, because it now has FDA approval. And so there are certainly people who are using it in the kind of adult motility space as sort of similar to how we would use an impedance study to kind of predict response to therapies and help guide treatment.
And this can only be performed in a sort of clinical service. Is there a sort of a home version that people can sit at home with?
So that's a great question. I've actually tried to send people home with this. It did not go well. It really works best. And what we've learned kind of from these preliminary data is you really have to be sitting still for 4.5 hours. The type of chair matters. So really it is very positionally dependent. And so that's why we found it works best in a clinical setting. And one of the really cool things that we've actually done at Nationwide Children's is we're actually using it in a CF clinic visit. And so people are getting the array, they're getting their physical exam, they're getting an array placed, and then everybody that needs to come and talk to the patient and ask them questions are actually doing it while they're sitting there in clinic on a day that they already plan to take off. And my hypothesis is that's something that's really going to improve the tolerability of doing this test.
Thank you.
Sorry. I was just wondering about the design of your study. What time of day were you taking these measurements, and was there a period of fasting prior to? Because,
yes.
Well, you probably know why.
Yes. So the test requires a 4-hour fast. And so I think almost exclusively have done these studies first thing in the morning. So people come fasting and then they eat the test meal. Usually by the time they get in, they get the meal. It's usually about 9:30 a.m.
Okay, thanks.
These are always my favorite questions.
Thanks for Sarah. Perhaps as the token diabetes doc in the room, I'm wondering what you think the effects of GLP-1 receptor agonists might be on gastric motility, particularly in CF, and if there's a role of considering, are there a subset of patients who might benefit more or less because of side effects to these new treatments?
Yeah, so I think that's a great question and something I've definitely thought about because my mentor is an endocrinologist. I don't know if you know that. But I think one of the really interesting things about GLP-1 is the side effects, the GI side effects of GLP-1s are very similar to the GI side effects of delayed gastric emptying. And I asked one of our obesity doctors sort of, do you think that that's the reason that people are actually eating less on these medications, or is there something else happening? And he couldn't really give me a straight answer. And so I think that we really need to actually use this device in patients that are treated with GLP-1s to better understand kind of the mechanism of those sort of early satiety symptoms and whether those are actually impacting weight loss with GLP-1s.
Wonderful presentation. Just wanted to find out, what about the cost?
That's a
What's
great
the
question
cost?
also.
Yeah.
So the costs, I've been using this kind of in varying settings for about 5 years, and the cost has definitely increased over those 5 years, as has the technology and support for the device. But now with FDA approval, there's actually insurances that are covering it and adults that are using it for clinical purposes.
All right. Thank you
you.
very much.
From the